All right, hi everyone, I'm David Pan, and today I'm going to be presenting on EQTL analysis. But first, of course, I'd like to thank our sponsor, the Computational Medicine Department at UCLA, for sponsoring these videos. All right, so first, we have to explain what the acronym EQTL stands for, which is Expression Quantitative Trait Loci. And I know that's complicated, and so we're going to try to unpack that. But essentially what we're trying to do is we're trying to use linear regression, which is explained in another video, to find the association between gene expression, hence the expression in EQTL, and a specific variant SNP or loci, hence the quantitative trait loci. And the real purpose of EQTL analysis is to look at the underlying genetic elements that explain the variation and differences in gene expression between individuals of a population. There's also another element of this where the genetics is really the heritable part of it, whereas gene expression varies day to day or individual to individual. The genetics are usually don't vary over an individual's lifetime, so this is really what we're trying to get at through EQTL analysis. So first, a little bit of review. As you've seen in other modules, DNA is double-stranded, and everyone has 2 copies of this DNA in the case of 2 chromosomes. And then these 2 copies of the DNA can have different alleles. So as you can see, for example, in position 2 here, there's one copy of the DNA has the allele T, and the other one has the allele A. And one of these is referred to as the reference, and the other one as the alternate. And over all these positions, as you can see, there's differences, and it doesn't necessarily have to be that there's just one single base pair. There can be multiple base pairs, as in position 7, or there can be insertions or deletions, as shown in position 14. So another way to describe these alleles, of course, as you've seen before, is ancestral and derived. And ancestral is, of course, the allele that's in the species before humans, which is usually primates or even further back. And then the derived one is the newest allele. And usually when we're talking about alleles, we have to talk about allele frequency. And this is also explained in another module. The math behind this is explained very well in the module on population genetics. But very simply, when we look at the alleles in the population, using the same example of TNA, you can see that in this population, there's at position 2, there's 6 T alleles and 4 A alleles. So that, of course, makes the frequencies for TNA 60% and 40% respectively. And therefore, we can now assign these 2 alleles another name, which are major and minor. So, of course, the major allele is the allele that's more prevalent in the population, and the minor allele is the one that's less prevalent. So usually when we represent these alleles, we usually like to represent them in terms of numbers instead of letters. So overall, in a file type that you've probably heard about before in these modules is a VCF file. We usually represent that as a chromosome, a position, and then we list the reference allele, the alternate allele, or in some cases the major and minor in that order, and then we'll indicate the number of alleles each individual has using a number 0, 1, or 2. And where 0 indicates that they have 2 of the reference allele, 1 represents the fact that they're heterozygous, so they have 1 of the reference allele and 1 of the alternate allele, and then a 2 representing that they're homozygous for the alternate allele. So usually the format of these is in a VCF file, which is explained in great depth in another video as well. But usually the way they're represented is all of these for every single variant. Sometimes they're divided up by chromosome for easier computational analysis, but sometimes it can be just one huge VCF file.